easier because in the end, there's less competition in these extreme environmental conditions. Okay, so spirulina farming these cyanobacteria, why are people interested in this? Well, basically it's quite popular as a food supplement. So we can buy this either as a powder usually or also pressed into tablets. And this is down to the properties of the cyanobacterium. So there's a high protein content, it has a lot of antioxidants and minerals. These are sort of the health benefits why spirulina, what spirulina is advertised for. People use it for all sorts of things. You will maybe find these spirulina smoothies. You can bake with it, as you can see here in these buns. Whatever you're interested in, I'm sure there is a way of incorporating algae into the food. In the end, we have to say that really this type of farming of cyanobacteria is already an established and serious application of these organisms. So just to give you an example here, this at the bottom corner, this is an example from an artisan spirulina farm in France. Typically, spirulina is grown in these sort of raceway ponds, and there is sort of this trend towards more smaller, local, fresh production of these organisms for direct consumption. In general, the global spirulina market is estimated at around $350 million in 2018, and the idea is that this is supposed to, I think, double by 2025 or something like that. So really, this is a serious market. There's a serious amount of money in this, and this is already established. So spirulina already took off. This is already happening, so to say. Okay, but this is an application, of course, of cyanobacteria, but maybe this doesn't really fit into what we would consider biotechnology yet. So this brings me sort of to the second part of the talk. So how do now cyanobacteria fit together with biotechnology? So if you think of microbial biotechnology, this is dominated by very advanced, very well-established heterotrophic chassis. This can be bacteria, for example, E. coli, bacillus, and all these other ones, or it can also be eukaryotic organisms, for example, yeasts. And this is obviously a sector that is established and things are up and running. They are improving dramatically at a quite ridiculous speed to some extent. So the question is, what do cyanobacteria have to do with this? How do cyanobacteria sort of fit into this picture of microbial biotechnology? Well, really, we have to think, this is sort of down to global developments, I would say. So this is an example here in this figure. You see the Sustainable Development Goals from the United Nations sort of summed up. And some of them might not be so relevant for microbial biotechnology, but others actually really are. And a lot of them, for example, here, affordable clean energy, climate action, or also here, life below water. There are several of them that really things have to change, and people are wanting them to change towards more sustainable systems, using renewable sources, creating a circular bioeconomy. This is obviously, there's a lot of ideas, how can we address this, and we can discuss, how do we get there? But the big question is, of course, how? And we think that maybe cyanobacteria could have their role in sort of moving towards these more sustainable systems and basically making our planet sort of advance further and use these organisms. So what is this down to? Why am I saying that cyanobacteria are relevant for this? So this is down to sort of very generically the perks of, I would say, green microbial chassis. So if you look here at these, what I call green microbial chassis, what you will notice is that I kind of lumped our cyanobacteria in together with also eukaryotic microalgae. So from an industrial point of view, when you read papers or when you look at these things and you read about spirulina, usually it will be referred to as a microalgae. And this is a general industrial thing. So when a biologist is asked, about cyanobacteria, they will say, oh, these are not microalgae. This is not a eukaryotic organism, which is true. But in an industrial sense, they are sort of one group, I would say. And this is basically down to the fact that to grow them also in an industrial setting, they will have very similar requirements, I would say. And this is down to their photoautotrophic growth. So what do we need in order to make these guys grow? Basically, they fix carbon from CO2, and they utilize sunlight basically as a free resource to turn this into chemical energy and to drive their metabolism. And this is the same for these, and that's why from a industrial point of view, they are kind of one group together. Compared to maybe other green chassis, what we also have to say, they are relatively easy and relatively fast to grow, which of course is attractive for anything that's biotechnologically relevant. And the additional thought is that also they can be cultivated on non-arable land. So really here, we're not potentially directly competing with land space for example, growing crops. Some of them will also grow in salt water, which is always nice. So for example, some of these marine species, or there have also been a lot of studies looking at growing these organisms in wastewater and sort of taking nutrients out of wastewater in the form of bioremediation. So this is sort of the general overview of what are the perks of these organisms. But now the question is, of course, what are they good for, which is here highlighted by the different types of products, I would say, that you could produce with these organisms. And to put this into a more general context, I took actually these examples here of potential target markets opportunities in Europe from a paper that we see referenced here at the bottom. And basically they say that there's 6 major ones: biofuels, bioplastics, biofertilizers, nutraceuticals, pharmaceuticals, and cosmetics. And I think this is probably quite representative in general for where microalgae and also cyanobacteria could play a role for a future application within biotechnology. Okay, so we have identified the potential and where we could use them, but now what is sort of the reality? What is already out there? And how can we move our cyanobacteria into biotechnology? And really this, there's already an example that I think goes into the right direction. So we're going back to spirulina. Spirulina is actually the only cyanobacterium that has a so-called GRAS status. GRAS means generally regarded as safe, and what it means is it's basically safe for food consumption. And this is a couple of years old already, but what you can see is that actually there was, for many, many years, the attempt was to actually find a natural blue colorant for food. And it turned out to be actually quite difficult to find something that's stable and that can be used, for example, here in sweets and confectionery. And of course, it's happy for me to say that the cyanobacteria could do the job and they can actually deliver a solution for this. So here, for example, from Christian Hansen, from the website, I stole this. You can see that these blue and also green colors in sweets, for example, they are actually produced by extracting pigments and proteins from spirulina. So what that means is we already have cyanobacteria moving into biotechnology, and really we're starting to see that there is potential and to start to use that. However, apart from spirulina, really there is not that much out there. Apart from that, we're still very much in the infancy. So the big question is, why is that the case? So what is sort of holding back this transition into industry? Because as we will see also further down in the presentation, cyanobacteria are interesting and they can do all sorts of things, and it has been shown with many proof of concepts. But what is sort of the problem? Why do we not see this popping up more in an actual industrial setting? So this is down to challenges and limitations that I want to discuss in a little bit more detail. To sort of figure out what is actually the problem, where does it start, I thought I will introduce this maybe a little bit differently. So if you go to Web of Science and you do a search for the topic of E. coli, basically what you get is that from 1945 until now, until 2020, you get around 500,000 entries, so 500,000 papers with the topic of E. coli. That is quite an impressive amount. And even from this amount of papers, there's around 13% which fall into the category of biotechnology and applied microbiology. So that is a serious amount of studies that have dealt with these problems and that have looked into how can we make and improve these systems. This is E. coli, so keep this in mind. If we now do this for cyanobacteria, so we have to think that we are actually comparing cyanobacteria, a whole phylum of organisms, with a single species. Yeah, and what we find is that there's around 35,500 entries for the topic cyanobacteria. And at least around 10% of that is actually for applied papers, biotechnology and also applied microbiology that it has been characterized at. So you can already see that this is a completely different dimension. So this is more than 10 times less than what you find for E. coli. And that's very, I think, illustrative of the whole problem that we're sort of facing. So if you think of us working with cyanobacteria as Team Green, and if we look at existing microbial chassis in biotechnology, really maybe, we can do the job if you're, for example, into cooking, I don't know, or something else, a hand whisk, it will do the job. But what we're really trying to compete with here are very advanced automated systems that have been, optimized over a long time. Maybe cooking is not your thing. You can use all sorts of other analogies here. Maybe a football team. So really our cyanobacteria clearly have potential. So these guys can kick a ball. But in order to compete in the Champions League, the question is, how do we get there? And at the moment, this is not very fair. So the question is, with what strategies, how can we be smart about basically advancing these guys here in a relatively small amount of time as far as we can, in order to be able to then compete here in the Champions League? And that is sort of the big question that we're trying to address and figure out. And obviously there's no easy solution to that, but there's ideas of how to go forward. In general, to sort of put these challenges and limitations into very broad terms of into what categories do they fall? Well, as I mentioned before, we have many proof of concept studies in the laboratory that show they have potential, that show what they can possibly do. But now what we need is sort of the transition into pilot scale, we need the transition into industrial scale and really into a proper scale up to see these systems translated into an industrial setting. And there will be many challenges along this way because these things are not linear, and what you see in the lab might not be reflected very well if you scale this up. So there's a lot of things that need to be addressed and we need to understand and figure out in more detail. Now, if we sort of put all these different challenges and limitations into individual categories, I think the first one, which is maybe most relevant to what we are interested in, are the biological ones. So of course we are often working with wild type strains, for example. We try to make them produce things, but really we need to understand these organisms better and we need to improve them to improve their productivity. Then there's obviously technical challenges. So actually transferring a system from here to the next scale and to the next scale, this is not so straightforward. This comes with process and chemical engineering problems that have to be solved and that are not so straightforward necessarily. If you think about it, for example, if you pick up a fermenter of E. coli that works brilliantly, you cannot just dump your cyanobacteria into it and expect it to work. You have to think we have very different conditions. So for example, here, they need sunlight. So we need to get the light somehow into these production systems. And that can be tricky, just to mention one challenge. So there's a lot of technical challenges that come with actually upscaling these systems. Now, something that we cannot deny, obviously this is also down to economic challenges. So if a system is not economically viable, then this is an issue and we have to figure out how can we make these systems economically viable? How can we produce these cheaper, or what are the way forwards to get there? And then maybe sort of tying a little bit into also to the other challenges, we also obviously have legal and societal challenges. So if you think of legal ones, for example, in Europe, we have quite strict regulations about genetically modified organisms, which we need to figure out how our cyanobacteria fit in there. And we also have to explain to society, what we are doing, what we're trying to do, what are maybe the advantages of these systems, but also what are the dangers and what are the risks and how are we addressing them to make sure that what we do is safe and is working for the purpose we're doing it. So this sort of ties all into this together. If you're a little bit interested in sort of these legal aspects, there's an article that I referenced here at the bottom. This is not only for cyanobacteria, this is actually mainly for microalgae, specifically in Europe, but this applies in the same way to cyanobacteria. So take a look if you're interested in that. And in general, this is obviously only a very short introduction and I cannot cover everything that is part of this, but I would be very interested to, during the discussion later on, maybe talk about this in a little bit more detail. So it would be interesting to also hear your thoughts. How can we address and overcome these challenges, and what do you think are maybe the most pressing ones of them that need to be solved first? Okay, of course there are challenges and limitations, but there have also been over the years very interesting and very important developments, I would say, to advance what we have. And I would say part of this is also the development of fast-growing strains. So I'm giving here this example of this Synechococcus elongatus strain, UTX 2973 from Himati Prakrasi's group. And this was published in 2015. And this strain basically is a freshwater strain. It grows only autotrophically. And the shortest doubling time that they've reported is 1.5 hours at 42 degrees. 1500 micro-Einstein of light, so quite a lot of light, and supplemented with 5% CO2, so also quite remarkable amounts of CO2. Why am I telling you this? If you think of 1.5 hours, we're getting into doubling times that are similar to heterotrophic organisms, such as, for example, certain yeast strains. So here we have doubling times that are seriously impressive, especially for a fully autotrophic organism, and this is where really things become more relevant also for biotechnology, because you have to grow these things in as least amount of time as possible, and if we can grow them in these time frames, things are obviously a lot more attractive compared to other heterotrophic organisms. Maybe very quickly about this organism, it's actually 99.8% identical to a quite well-known model organism, Synechococcus elongatus PCC7942. This is useful because if we want to advance the system further, we need to understand this organism. And that means that really, although this is maybe newly characterized, we already have a better idea of its biology than we would starting with a completely new strain. We have to say that. And there are some disadvantages of this particular one. So it's, for example, not naturally competent, as most of the model organisms that we usually use are. However, there have been several tools also developed to engineer these guys, and things are advancing. So yeah, you cannot get everything usually. Okay, this was just basically the first example, but since then there have been several further fast-growing strains that have been characterized over the years. There's also 2 other Synechococcus elongatus, one's PCC11801 and 11802, and we're not going to go into much detail, but basically they're all kind of similar, but then under certain growth conditions they're better than the other ones. So depending on what exactly your setup is, it might be interesting to sort of see what sort of fits best with this. So for example, this guy here grows faster, but then it needs higher CO2 than this guy and the other way around. So depending on what you're exactly looking for, it's nice that we're starting to actually have sort of a broader choice to go from. Then the last one, which was published in 2020 now, just recently, is Synechococcus PCC11901. This is actually a marine strain, so this is interesting because the other ones are freshwater ones, and it's actually similar to PCC7002. And it can accumulate a dry weight of 33 grams per liter in batch culture, with quite some CO2 supplemented. But this is quite a serious amount of biomass that you can grow from this strain, so that's quite extraordinary. And these are sort of the studies that are going into the right direction. Yeah? So here we're starting to see that we can actually grow these things quite fast, quite simply, and actually get serious amounts of biomass out of it. Okay, so basically this is the end of the idea of going from, we have challenges in biotechnology, but how can we address them in general? As a first aspect, we can, to start with, use organisms that grow faster. But really, as I told you, we're trying to compete in the Champions League, and we need to get there as fast as possible. And we're not going to get there if we don't use our own personal tricks to get there. And this is down to basically the third topic I would like to introduce to you, and that is synthetic biology. So I really think that through synthetic biology, we can really sort of advance these things faster and in a more dedicated way, specifically targeting a product, for example, to make these systems more effective in a shorter amount of time. I thought maybe very briefly, I will introduce sort of the core idea of synthetic biology. This is obviously a big field, and it's difficult to sort of just put this into a box. But very basically and very generically, I would say synthetic biology is sort of when biology meets engineering. So the idea of using principles from engineering to actually advance biology. So I'm sure you might have seen these cycles that are usually shown, where really this shows this iterative approach to advance a system. And this is down to designing something first, then building and testing it. And then also importantly, also learning from what you've built and what the test results were, to then go through in an iterative approach next round of improving the system further through sort of this cycles of improving something towards a certain goal. I would say that synthetic biology has majorly been sort of boosted by advances in DNA sequencing and also DNA synthesis, mixed with basically much more advanced bioinformatic tools and high throughput methods. In general, the idea of this engineering component is more this sort of rational design to construct a biological system from individual parts. But what is really important is that this is also quite open community, and really there's a big effort towards standardizing these individual parts that you can then pick up and you know, transfer it into a different system. So this is this idea of a modular standardization of parts, and we'll actually get back to that in a second again. So this is obviously nice. I mean, this is synthetic biology, but what does this have to do with cyanobacteria, and how could it actually help? First of all, I want to just briefly maybe point out also something we have to keep in mind, that these are all nice ideas and we would like to use them. However, there's also certain limitations to actually use synthetic biology at its full options in cyanobacteria. And what I'm mentioning here actually is not necessarily limited to cyanobacteria, but it is also the case for them. So if we look at this cycle here, in each of these stages, essentially there are certain, I would say, challenges that will basically kill a project if this is not given. So if you look at the design part, so for example, if you want to integrate or if you want to modify an organism and you do not have a genome sequence, well, it's not really going to be possible. How are you going to know where in the genome to integrate your construct or something like that? So really you need a genome sequence and it needs to be annotated in order to start designing something. Then in the build phase, same problem. So if you do not have a promoter that you know works in this organism, if you do not have genetic tools in general, no markers maybe, or you also do not have a system in order to transform this organism, well, then you're not going to be able to engineer it, and then basically the project stops here. Concerning the test phase, well, specifically for cyanobacteria, I would say compared to other heterotrophic organisms, quite often I would say we're quite behind in the amount of screening and high throughput methods that we actually have available for us. And there have been several studies addressing this. For example, if you look for a recent paper from Paul Hudson's group looking at pooled CRISPR-I libraries in order to screen for enhanced industrial phenotypes, things like that. These are really important developments, but we need more of that, and we don't have that much of that yet. And in the end, if we get to the learn stage, well, learning something, maybe you might think that this is limited to what you know about something, but it might not be limited to what you know. It might be limited to what we know as a community about these organisms. So we do need a general understanding of the physiology and also of the metabolism of these organisms. So basically what we need is we need the cellular context to interpret our results. And that can sometimes be quite limiting in cyanobacteria. We just don't know as much as we know, for example, for E. coli. Good. However, what that means is, as a conclusion from this, is synthetic biology will be limited to some extent to organisms that are well characterized, which in most of the cases will be model cyanobacteria that are genetically tractable. So this is something we just have to keep in mind. So whenever we try to use these things, we're kind of a little bit limited, although this is getting better to what we already know and that we've already studied in more detail. I mentioned earlier that part of synthetic biology is sort of this really big idea of standardizing things and making them be useful across systems and across labs, for example. And there's many examples where kits and tools have been developed for different cyanobacteria, and I cannot mention them all, but I wanted to highlight this one here from 2019. It's called Cyanogate, and this is very much in the synthetic biology realm of things. So this Cyanogate is a Moklo system. It's a Golden Gate system that is based on the plant Moklo syntax, which means that then it's easier to actually share parts between these different systems and to transfer them. And basically what you do is you have your individual parts here in level 0, for example, a promoter or a gene of interest, and then you assemble them in level 1 into transcriptional units. And then these transcriptional units can be put together in level T. And then depending on what you're interested in, there's different level T backbones to either integrate it or to have self-replicative plasmids to transform into your cyanobacterium of interest. And there's actually tools for several different cyanobacteria. So if you're interested, this is also available on Argene, and this kit has been developed by Alistair McCormick's lab in Edinburgh. Okay, so now we maybe have an idea of what synthetic biology is, and now the question is, what are sort of the examples where synthetic biology has been used or applied to sort of improve cyanobacterial chassis? And here I clearly really have to say that I cannot do justice to all this work that has been done. There have been some really great studies on how to do this and showing what cyanobacteria can do, what kind of potential they have. But I would say in general, if we look at an overview of products that are interesting from these organisms, we can separate them into commodities and fuels. So for example, many alcohols, alkanes, alkenes have been produced, sugars, or also hydrogen. If we look at more high-value compounds, people have expressed recombinant proteins, recombinant enzymes, natural products. And natural products is maybe more of a niche thing in the sense of that cyanobacteria have natively sometimes very interesting natural products, but this can be limited to actually understanding their biosynthesis itself. So quite often when we talk about high-value compounds, these are sort of secondary metabolites, for example, from plants that have been characterized in other organisms. So for example, isoprenoids or phenylpropanoids, just to give an example. Also pigments. So obviously cyanobacteria have pigments natively, but then we can also, as we will see, engineer their native metabolism towards other pigments that they might not naturally produce. So in the end, as you can see here, synthetic biology, biotechnology, how this all fits together for cyanobacteria is majorly down to actually engineering these guys and improving them. And as I said, I cannot do justice to all this work that has been done, but what I was thinking is I would give a couple of examples of very, very recent papers where they have been employing sort of these strategies to make cyanobacteria more effective chassis. The first example I wanted to give is about a pigment. So this is astaxanthin, and actually in 6803, this is a typical model organism that you've probably heard a lot about by now. Basically, astaxanthin is not naturally produced in Synbio. What Synbio does produce, however, is beta-carotene, which is a precursor of astaxanthin. So what they did in this study is they basically expressed hydrolase and the catalase to then add in the biosynthetic pathway to get from beta-carotene via canthaxanthin and zeaxanthin down to astaxanthin and produce it. And what is quite impressive about this study is more the amount of work they went through to then, in iterative cycles, improve the system to get rid of bottlenecks, for example, here to have beta-carotene availability, and to then also go further up into central carbon metabolism and MEP pathway, for instance, here, in order to improve that system. And I think by the end in the paper, they were at strain number 29 or something, where they did steps of improving and testing different enzymes to get these cyanobacteria to really produce a lot of astaxanthin. So take a look. This is a nice recent example of metabolic engineering in cyanobacteria. So the second example is maybe a little bit different, and maybe I really wanted to show this because I think it's really important that we start thinking a little bit out of the box about the issues that we're having and how we can address them. And basically, this is from Patrick Jones' lab at Imperial College London. And what they've been doing is they looked at derivatizing products. So they did this with octanol, I think. So basically, this is the product here that we would be interested in to produce. But what they suggest is that actually, like within the cell, we can engineer the cell in order to derivatize this product of interest to make it less toxic, more or less soluble to the cell, and make it therefore be different and not available maybe for the general metabolism of the cell. So the idea is, for example, you can make it more hydrophilic by glycosylating it, or more hydrophobic through esterification. And really the idea here is to make the biology inside the cell to work together with the chemistry then later on, so that basically we derivatize the product in the cell to make it less toxic, and then be also able to accumulate at higher levels inside the cell. And then afterwards, what we can do is we can use chemistry to then remove this derivatization and basically get the product that we were actually interested out. So I think this is a nice idea of how we need to think about, how can we improve our systems from a bit of a different angle. And finally, I have one more example I wanted to show you, and this is really more a general principle, and it's called light-driven catalysis, and this has been developed starting in around Paul Jensen in Copenhagen and colleagues. And basically the idea here of light-driven catalysis is that actually we can use photosynthesis essentially, of course, to produce something. But actually we can not indirectly only use photosynthesis because we needed to actually for the cell to live and to build up biomass and everything, but we can directly also use these electrons from photosynthesis. So here we have, for example, photosystem 2, photosystem 1. This is the photosynthetic electron transport chain. And basically there's a lot of electrons that sort of go through the system, and then they have to be taken care of. And the idea is that we can directly use these electrons to supply them to redox enzymes, such as, for example, this P450 here, in order to drive catalysis of these enzymes. And there's different examples, so this doesn't have to be from the photosynthetic electron transport chain. This can also be, for example, through a photosensitizer. But in the end, we sort of kind of try to show different categories of light-driven catalysis, and this is explained in this review in a little bit more detail. What I want to do is give you this example here in A in a little bit more detail. So basically the idea is that we have photosynthesis that is operating in the cell anyway in the thylakoid membranes here. And what we can do is basically we can express cytochrome P450s. And usually what they need is they need an oxidoreductase, and this oxidoreductase will feed it the electrons for catalysis, for example, here from tyrosine, through essentially getting them from NADH and NADPH. And this step from the oxidoreductase to P450 can be very limiting. But when we now heterologously express these P450s in the thylakoid membrane, what we basically get is that we get these electrons from photosynthesis, and we can feed them to the P450s for substrate turnover. So this is sort of the general idea of this. And the first paper actually started with only one enzyme. This is CYP79 from the Durin pathway in Synechococcus. But then later on, as you see here in the picture, the whole pathway was also transferred in the Synechocystis, where we have 2 P450s and 1 UGT to actually produce Durin from tyrosine in vivo, using basically the energy from photosynthesis to drive these enzymes. Why am I showing you this? Well, what I would like to do is just very briefly also touch upon how this sort of fits into the work that we are doing. So coming from Paul Jensen's lab, I was quite interested in this, and just sort of still looking into these light-driven catalysis systems and how can we actually improve them further. And part of that is that we're also doing some prospecting to look at good model pathways. Sometimes there can be a lot of toxic intermediates, and if you want to do high screening or something like that, this might not be the best option. So for example, Sayali that you see here, she's working on sort of figuring out if we can establish some nice model pathways in our cyanobacteria to study this. And part of this is also, a lot of these enzymes are membrane bound. So really we have to understand how can we, sometimes proteins from very different organisms, how can we reliably target these proteins into the membranes, into the thylakoid membranes usually of cyanobacteria? And how can we basically employ metabolic and genetic engineering to then integrate this pathway further into the central metabolism of this of these cyanobacteria, and how can we sort of funnel the energy into the product that we're interested in? And there's also Annika and Dennis, they're also working on this, more on these two topics. So all of them will be attending the conference, and I'm sure they will be happy to talk to you about their projects in more detail. So that is about improving light-driven catalysis, but we're also interested in sort of the general idea of how can we achieve efficient metabolic channeling. So it's not necessarily about a specific product. So I'm not a company that wants to produce as much as possible of one compound. For me, it's more about how can we find principles in general to make these cyanobacteria be more attractive and be better at producing something. So if you think in general of an enzymatic pathway, usually it involves several enzymes, and that means that they have to work together. We have intermediates that form that have to find their way from one to the next enzyme, and you all know there's a lot that can go wrong along the way. So the idea is that we should maybe consider in more detail how can we maybe make these things work more efficiently? How can we build metabolic units and achieve some form of metabolic channeling? This usually also comes with a detoxification, because that means that if you have some intermediates that are toxic, for example, well, if you all basically put your enzymes into the same subcellular compartment, well, then these intermediates will not show up as much, and it will be actually better for the cell. So in the end, the idea is to co-localize and to build sort of these metabolic units for more efficient product synthesis. This can be, like I said, by encapsulating it, or it can also be by just providing a scaffold where the enzymes of a specific pathway can attach to come in closer localization to achieve sort of similar effects without having to get the substrate sort of into a compartment and then getting the product out. Okay, this is just sort of the general overview. Here, specifically the scaffolding project, actually my PhD student Alexandra is working on that quite a bit. So if you're interested in that, you can ask her about what she's been doing and where we are with that. One more last thing I wanted to mention is that this goes very much into what I said about the problem with the learn phase of synthetic biology in cyanobacteria. So as I said, we're really interested in making sure that we get our enzymes into a certain specific subcellular localization, but how do we achieve this? And we're kind of very limited to some extent quite often by understanding the underlying cellular mechanisms of how we can target something to a subcellular compartment. And for example, here for protein translocation, we know what systems operate in these organisms, but we don't really know how they make sure that, for example, something goes into the periplasm or something goes into the thylakoid lumen. So we're really also interested from a fundamental point of view of how, how things work in these organisms to then advance this in our applications. So this brings me to my take-home messages, I guess. In the end, basically what I was trying to tell you is that, as you can see, there are still multiple challenges and limitations that are kind of holding back the transfer of cyanobacteria into an industrial setting. But there is also already studies that show that things are working. So we have an implementation on an industrial scale of cyanobacteria. We have a good example. We have spirulina. Then there is interesting trends that I think are important to advance us as a community, which is, for example, these fast-growing strains. Then there is many proof of concept studies that really show what cyanobacteria can do, but really now what we need to do is sort of take this to the next level to actually implement this further. And I think to do that, quite often synthetic biology concepts can actually aid this process to make our systems improve also on a shorter time scale. So in a nutshell, what I hope that you take away from this is that cyanobacteria do have an interesting potential for biotechnology, specifically for sustainable biotechnology, and we're only sort of at the starting point to explore also how on a larger scale we can sort of implement the systems, what are the challenges and how can we implement them. So really what I'm trying to say is I think they deserve further attention, and it's very nice that I got an opportunity to talk sort of to the new generation also about this here at the conference. And we really need more people interested in that and working on this to advance these things faster. So as a closing question, this goes really to the audience here. I would be quite interested in hearing what you actually think. You know, do you see that synthetic biology can make it happen or not? So yeah, Penny, for your thoughts. I would be really nice to discuss this in more detail. And these are my acknowledgements. So these are people that I'm currently actively working with. And apart from them, of course, mainly my group, I would also like to thank you for listening to me and for following me through this presentation. And if you don't catch me or my group at the conference, get in touch if you're interested, be it by Twitter, by email, or in any other possible way. So thank you for your attention.